Good afternoon. My name is Joseph Marino. This is my 10th grade biology educational presentation, Transport Across Cell Membranes. Understanding the fundamental mechanisms that enable cellular processes is crucial for sustaining cellular homeostasis. The transportation of molecules across cellular membranes is a key mechanism that plays a critical role in this regard. Among these processes, osmosis plays a particularly significant role in regulating the hydration of cells, which is crucial for their survival and optimal functioning. Active and passive transport, tonicity, and osmosis sustain homeostasis by moving soluble molecules across lipid membranes of living organisms. By comprehending these mechanisms, we can maintain health and well-being by ensuring that cells function optimally and conduct essential metabolic processes while maintaining their structural integrity. The cell membrane, or the plasma membrane, is a crucial component of all living cells. It is a semi-permeable membrane that surrounds the cell and separates its internal environment from the external environment. The plasma membrane is composed of a complex structure that includes two layers of phospholipids, which are arranged in a way that creates a bilayer. Here's the phospholipids, here's one layer, and two layers creating the bilayer. Each phospholipid molecule has a hydrophilic, water-loving head and a hydrophobic, water-fearing tail. The hydrophilic heads are oriented towards the aqueous environments, either outside or inside the cell. The hydrophobic tails are oriented towards each other and away from aqueous environment. Here are the heads, here are the tails, and they're meeting each other. This unique membrane structure enables it to selectively regulate the entry and exit of substances into and out of the cell. This bilayer provides a barrier to the free flow of molecules, making the membrane semi-permeable. Apart from phospholipids, the plasma membrane also contains proteins, peripheral proteins, channel proteins, globule proteins, glycoproteins that are attached to the surface of the membrane. These proteins play various roles, such as transporting molecules across the membrane. They also help facilitate cell-to-cell communication and provide structural support to the membrane. Channel proteins are integral protein components, forming aqueous channels through the lipid matrix. They facilitate the selective passage of ions or molecules adhering to their concentration gradients. These proteins may be gated or ungated, modulating substance flow in response to diverse stimuli. Cholesterol is also present in the plasma membrane. It helps to maintain the fluidity and stability of the membrane and is essential for the proper functioning of the cell. Membrane transport. Movement of molecules through a membrane. Passive transport. No ATP required, meaning no energy. Simple diffusion, facilitated diffusion, and osmosis. Passive transport. Movement from a higher concentration to lower. No energy, no ATP. No carrier protein required. Very small O2 to CO2 lipid soluble molecules, steroids, can be transported. Passive transport is a type of cellular transport that occurs without the need for this energy. In this process, molecules move from an area of higher concentration to an area of lower concentration. Unlike active transport, which requires energy and carrier proteins to move molecules across the cell membrane, passive transport occurs spontaneously. However, it's important to note that not all molecules can be transported through passive transport. Only very small lipid-soluble molecules like oxygen, carbon dioxide, and steroids can diffuse across the cell membrane. These molecules move through the phospholipid bilayer of the cell membrane, which is composed of the hydrophobic and hydrophilic tails that repel water and allow small nonpolar molecules to pass through. Passive transport is crucial for the process in maintaining the proper balance of molecules within the cells and across the cell membranes. It allows cells to obtain the nutrients they need to function properly and to eliminate waste products that could be harmful if they accumulate. You have diffusion, cell membrane permeability, molecules scatter because they collide, substances move down the gradient without energy. Facilitated diffusion, like passive diffusion, involves the movement of molecules in a direction determined by their concentrations inside and outside of the cell. Molecules that are polar and charged, i.e., carbohydrates, amino acids, nucleosides, and ions, encounter obstacles when attempting to cross the membrane due to their hydrophobic core. While water molecules are small, they lack the full charge result in slow movement across the membrane. Carrier proteins offer a means of transportation across the membrane by selectively binding to specific molecules and undergoing conformational changes. The process of facilitated diffusion is fueled by the concentration gradient, which causes the molecules to flow from regions of high concentration to those of lower concentration. So you have high concentration to lower concentration. Here's the carrier protein, and then it diffuses and comes through. It should be emphasized that this process is passive, meaning it doesn't require any cellular energy to occur. Carrier proteins play a critical role in this process as they facilitate the movement of various substances across the plasma membrane. For instance, glucose transporters enable the transport of glucose into cells, which serves as a crucial source of metabolic energy. Additionally, ion channels allow various ions, such as sodium and potassium, to traverse the membrane, while aquaporins enhance the movement of water through the membrane. Factors that affect the rate of diffusion. The rate of diffusion of a substance depends on various factors, such as the concentration gradient, temperature, and the size of the substance. The greater the difference in the concentrations of a substance, the greater the rate of diffusion. As temperature increases, the rate of diffusion increases. However, as the size of the substance increases, the rate of diffusion decreases. Then you have solvent properties. You have viscosity and density. They significantly affect diffusion. If the medium, the solvent, through the particles diffuse is dense or viscous, diffusion becomes more challenging. These factors play a crucial role in determining the rate of diffusion of a substance. Types of active transport requires the breakdown of ATP for energy, primary active transport, P-type ATPase. Secondary transport utilizes electrochemical gradient established by active transport and the sodium glucose transport proteins. Active transport requires energy, usually in the form of ATP, to move molecules against their concentration gradient from an area of low concentration to an area of high concentration. P-type ATPase is a type of protein that actually transport ions such as sodium and potassium across the membrane. Secondary active transport utilizes the electrochemical gradient established by primary active transport. Sodium glucose transport proteins are transporters that move glucose into cells along with sodium ions. Primary active transport. ATP hydrolysis. You have your three phosphate groups. You have your hydrogen, your oxygen. Okay, breaking the high energy bonds that converts chemical energy to mechanical energy. Substances can move from a low concentration to a high concentration. The net flow of molecules by facilitated diffusion through either carrier protein or protein channels is always energetically downhill in the direction determined by the electrochemical gradients across the membrane. In many cases, the cell must transport molecules against their concentration gradients. In active transport, energy provided by another coupled reactant, such as hydrolysis of the ATP, is used to drive the uphill transport of molecules in an energetically unfavorable direction. Examples of primary active transport. Ion pumps are crucial for maintaining ion gradients across the plasma membrane. The NAKATPase, sodium potassium ATPase, for instance, uses energy from ATP hydrolysis to transport sodium and potassium against the electrochemical gradients. Sodium concentration is higher outside the cells, while potassium concentration is higher inside the cells. Potassium is inside the cells, where sodium is outside the cells. Greater concentration outside and a lesser concentration of potassium inside. The pump transports these ions by undergoing ATP-driven conformational changes. Initially, Na ions bind to high affinity sites inside the cell, prompting hydrolysis of ATP and phosphorylation of the pump, leading to conformational change. This change exposes the sodium binding sites to the outside of the cell and lowers their affinity for sodium, resulting in the bound sodium being released into the extracellular fluid. Simultaneously, high affinity potassium binding sites appear on the cell surface. Extracellular potassium then binds to these sites and stimulates the hydrolysis of the phosphate group bound to the pump, inducing a second conformational change. This change exposes the potassium binding sites to the cytosol and lowers their binding affinity, so potassium is released inside each cell. Each cycle of the pump transports 3 sodium and 2 potassiums across the membrane. So you have 2 potassiums and you have 3 sodiums using 1 molecule of ATP. 1 molecule of ATP produces 2 potassiums and 3 sodiums. Osmosis. Osmosis and diffusion are both physical processes that involve the movement of molecules. However, osmosis is a specific type of diffusion that occurs only in fluids. Unlike diffusion, which is present in solids, liquids, and gases, osmosis involves the selective passage of water molecules through a semipermeable membrane. This means that larger molecules are unable to pass through the membrane. Osmotic movement happens from a region of high water concentration to a region of low water concentration until a state of equilibrium is achieved. Nature's solution is dilution. This is the concentrated sugar and water. Okay, here's your sugar molecules from a high concentration to a low concentration. Here's your selective permeable membrane. Okay, and then here's the same thing. You have a high concentration and a low concentration. Okay, and then it's moving towards the solutes. Plants also rely on osmosis to absorb water and vital nutrients from the soil. While in animals, including humans, osmosis is a key process used by, by the kidneys to filter waste products and maintain proper fluid balance. By exerting pressure on a solution, osmosis can be halted. This pressure is known as osmotic pressure. Osmotic pressure varies depending on concentration and temperature, behaving similarly to gases. Osmosis plays a critical role in biological processes such as nutrient absorption, waste removal, and maintaining cell turgor pressure. Excuse me. Additionally, in technology, reverse osmosis utilizes this principle by forcing the solvent, which is usually water, to move against the concentration gradient, filtering out any impurities. Remember, osmosis is like nature's way of balancing the scales. Water moves where it's needed most. And again, nature's solution is dilution. If the pressure exerted, osmotic pressure, the pressure exerted by the molecules on one side of the membrane is greater than the pressure exerted by the solute particles on the other side, the process of filtration takes place. Filtration is the movement of water back across the membrane due to hydrostatic pressure, which can stop the process of osmosis. You have the semi-impermeable membrane, you have the osmosis, it's moving to the right, water shifts to the right by osmosis, where the water is higher concentration of solutes, the pressure of gravity is on the left side. Tonicity. Tonicity refers to how an extracellular solution impacts a cell's volume by affecting osmosis. Solutes, which are solid substances like salt or sugar, dissolve in a solvent, usually water in living systems. Isotonic solutions have the same soluble concentration in cells in an isotonic environment, experience water movement without any changes to their biology. Hypertonic solutions have a high solute concentration relative to cells, causing a net water movement from the cell's high water concentration environment to the outside, resulting in cell shrinkage. Then you have hypotonic solutions, which have a high low solute concentration, causing a net water movement into the cell, causing cell swelling. This is the hypertonic. The water is coming out, which is causing it to crenate, which is causing it to shrink. This is the isotonic, where it's got a state of equilibrium for the water that's going inside the cell and the water that's going outside the cell. And then this is the hemolysis, okay, where it's being broken, okay, and the fluid is going into the cell, which is causing swelling. Conclusion: diffusion, passive transport, active transport, osmosis, and tonicity. Thank you for your attention during my presentation on cell membrane and transport processes. I hope you found the following concepts helpful: diffusion, passive transport, active transport, osmosis, and tonicity. Diffusion is the movement of molecules from a high concentration to low concentration, where passive transport does not require energy, while active transport requires energy to move molecules against the concentration gradient. Osmosis is the movement of water molecules from high to low concentration. Tonicity predicts the direction of water movement across the membrane. These processes are critical for maintaining physiological balance, regulating the concentrations of various substances, and preserving the cell's internal environment. Thank you for listening. I hope you found this information valuable. Have a great night and God bless. And remember, nature's solution is dilution.